Hi everyone, I'm Umran Yaman. I'm a PhD student in Hardy Group at UKDRI at UCL. Today I will be presenting part of my PhD project, but before I would like to thank the organizing committee for this great opportunity. Okay, so my title is quite long. Synaptic adaptation via microglia, isoform alterations in APP knock-in mouse model of Alzheimer's disease detected by long-read analysis. So, genome-wide association studies highlights the risk variance mainly expressed by microglia, but how those act on the transcriptome or genome is not clear, especially when SNPs are not changing the protein sequence, and we cannot predict them easily as well. Initially, we wanted to get an opinion about their possible splicing effects. And for that specifically, we have used APPNL-G-F mouse model. So we have had 6 control, 6 mutant, and we have controlled for the batch effects and gender effects as well. We have had 9 months of age mice, and we have used bulk cortex. And to accurately detect the splicing, we needed to create a full assembly of the isoforms, and we have used Oxford nanopore cDNA sequencing. So we started with the splicing, aiming detecting splicing, but we have performed different level of analysis. So we have started with the gene level analysis, and I can show here, I think. Yeah, so we started with the gene level analysis. As you know, that from the literature and the short read analysis, short read sequencing, we only have this information, only gene level information. We don't have transcript level information, mainly measuring isoform proportions and isoform switch, which I will go in a bit later. And we did also perform exon level analysis, which measures specific genomic locations compared to other locations. So from gene level to exon level, we have quite comprehensive data with APPNL-G-F mouse model. And finally, we performed splicing. Splicing is quite complex, so we have identified many events in APP knock-in mouse model, including exon skipping, intron retention, alternative splice usages. This is a summary of what we have performed. I will briefly mention on the gene level analysis, then we'll go into these. So short-read sequencing has limitations to fully identify the isoforms because of the sequencing gaps. We don't have much information about especially longer genes because the mapping quality might be affected and sequencing depth might be affecting the full assembly. For that, we have used long read, and we do have more comprehensive information about all the genes. This is a genome-wide study. On the right-hand side, you can see one example is APOE. We have detected 2 novel variants. It's 2 novel transcript variants within APOE in APP knock-in mouse model and controls as well. I have used Oxford Nanopore's pipeline, and I combined with the functional isotranscriptomics framework what those isoform is actually doing, whether they are real, whether they are protein coding, etc. And we have also created using different tools. Those are used to identify protein domains, coding potential, again, signal peptides, and whether there's any disorder regions within the novel and known isoforms. And I have used isoform switch analyzer to also detect the proportions or the relationships between the isoforms. If one isoform is up, whether another one is down, etc. So our results validates the short-read RNA sequencing. We have obtained amyloid-responsive microglial module. This is just a validation check. This is gene-level experiment. And the data on the left-hand side is single cell, while on the right-hand side, our long-read data is bulk level. And Yeah, long read as well. It's the different sequencing, but we get the similar enriched module. This is gene level again. This is a validation, different level of validation. We have get similar genes on the gene level, probably due to proliferation and activation. Again, since this is bulk, we cannot control for the proliferated cells or the cell numbers in general. So this is a new information. Again, differentially expressed isoforms. I haven't started usage yet. Here, specific isoforms are coming up, which are represented by the gene names. For instance, CCL6 and ThyroBP or TREM2. Those are specific isoforms, and on the right-hand side, as you can see, the second isoform is specifically up in the disease, and this is represented usually as one gene, and we didn't have this isoform information, and we do have genome-wide version of it right now. So in summary, on the expression level, gene level, and isoform level are quite similar, but we do have more specific or detailed version of the gene level analysis now. Now I will start to quickly present this. So this is a transcript usage results. With the transcript usage results, since they are the measure of specific isoforms to other isoforms, these are giving a different signals from the different cell types. So now we are seeing more neuronal pathways. Our top hits, for instance, synaptic green 1, which is a synaptic membrane protein which is responsible for trafficking synaptic vesicles. And we do get these genome annotations mainly enriched related to neuronal development and neuron differentiation. It's quite different than the expression level. On isoform level, it is telling us that it is not only about the microglia at 9 months of age in APP knock-in mouse model. There are different cell types playing role in disease or simultaneously with this microglial activation, which I will come in a bit as well. So specific transcripts in synaptic green is up, and those are not detected by differentially expressed analysis. Synaptic green 1, 202 specifically up in response to amyloid accumulation. And this switch is similar to transcript usage, but now we are looking at these inverse relationships between the isoforms, while one is down in the amyloid accumulation, and one is up. So this can give information about the protein changes as well. And here on the right-hand side, you can see just a subset of the list. We have detected cathepsins. SYNGR is one of the genes that we have detected as well. And we have detected novel isoforms. And again, the cell type enrichments is pointing quite different cell types. Again, not only microglia. That I have done the cell type enrichment that I will show at the end. So this is a different gene, CAPG, capin actin protein. This is disease-associated microglial gene and also has expression in the AD human. So we did have that information on the gene level. Now I am showing you more specific information about the CAPG, which isoforms there is up, and whether there is any patterns that we cannot see with the isoform usage. Actually, there is first isoform is basically up in control, and the second one, the other one is up. So there is an isoform switch, and we have detected novel transcript within this gene, and it's actually relevant to the amyloid accumulation. We have created predicted protein database in APPNL-G-F, and so these are basically based on the isoform sequences. And we have done also poly-A signal or polyadenylation site usage. Again, may inform us about the protein sequences or peptides that is derived from novel isoform or splicing. So we have that database as well. As I said, initially we wanted to check whether GWAS or familial or Alzheimer's disease risk genes affecting splicing, but we did not see any isoform alterations or splicing, which was quite interesting as well. We have checked a panel of Alzheimer's disease risk genes. Again, there is no splicing with the genes that we have checked. So transcript usage takes place in the multiple cell types. There is not much of sex differences observed. There's a slight differences. I can give more information about that if you're interested in. The panel of GWAS hits did not have this isoform expression in amyloid or isoform alterations in amyloid. We have detected novel isoforms and novel switch events. We have created this comprehensive database, isoforms, their protein predictions, polyadenylation usage site, which may inform us about the proteins as well. So exon-based analysis is also pointing out a similar list. So now from the gene level, we can specify which exons are specifically used that is deriving, so that is affecting the transcriptomics results at the end. This can be helpful for designing experiments and also can inform us if the protein sequence is changing, how this changes, whether which exon is affecting that type of change. Splicing-based analysis, so I have used PCI sigma. This is tailored for the long-read RNA sequencing, and also can perform with the short-read RNA sequencing, so we can compare to different technologies as well. We have detected clafrins, catepsins, and other again, other genes seems to be neuronal and also different cell types. But it will be, I guess, better if I can show the summary slide. So on gene level, we get this microglial enrichment in response to amyloid pathology. As I show in the first slide, that we only get these modules in bulk and single cell as well. On isoform level, including the isoform expression level, we get more specific information because we do annotate the individual isoforms. And we have got the T-cell related signals and also microglia and macrophages, while splicing is pointing out both T-cells and oligodendrocytes and some neuronal signals, again, an ependymal, for instance. It was interesting to see this enrichment. And isoform switch is, again, similar to usage, but is giving us this proportional change between the isoforms. It's a pattern within the isoforms, and it is giving us these neuronal cell types and astrocytes or T cells again, and usage is giving us about different cell types. So the take-out message from here is the differential gene expression analysis itself may not point out changes because it might be biased due to the proliferation, especially in the bulk tissue. And this type of information, the accurate identification of this can be achieved with the long read, and we have created this extensive database for your future, or a different type of experiments, and these analyses can be done within the different disease contexts and can be used in different applications or experimental designs. So we started with the hypothesis whether familial or Alzheimer's risk genes affect splicing, and interestingly, we did not find that. So now we hypothesize that this synaptic enrichment or synaptic adaptation, because we don't have the neuronal depth. So we think that synaptic adaptation by splicing is downstream of the risk variance, and this might be due to the synaptic protection at that stage, and in animal or in mouse model, then this neuronal death or tau in humans, tau pathology occurs after this stage. So With that, I would like to thank all the contributors to this study, but specifically I would like to thank my PI David Sali for all his support, and John Hardy and Francis Weissman for their support all the way my PhD journey. And thanks for listening.
Thank you very much, Umran. Any questions, please? So maybe I could start by just asking you to elaborate on what sex differences you found.
Yeah, there's like just 3-4 genes coming up with the differential transcript usage. So we have performed mutant versus mice and male versus female, and we get small list of genes, and we controlled for that. But I just want to.
But there's small changes.
There is small changes.
Okay,
yeah.
Thank you. Any other question? Yeah, there's a question over. 2 questions here, please.
Hi, it's really cool to see long read stuff, always. I just wondered a couple things, and I appreciate that you might have not done that kind of analysis, but you mentioned a couple times that you're finding some novel isoforms. So I was wondering overall, sort of like how many novel isoforms you're finding in your data sets and things like that?
Yep. As you may know, string tie shows us around 15%, 20% of the novel isoforms. Yeah, so we checked the precision or the sensitivity or the mapping qualities of those. So those seems to be consistent. There is around 15%. It's quite a lot of, as you also observed, yeah.
And I was also wondering in terms of your sort of cutoff points for what you are counting as an isoform, if you will, because obviously, like, there's quite a lot of debate in the field.
Yeah.
So, like, what are we counting in terms of expression levels, in terms of detection in the sample? So I was just wondering about that.
Yeah, it's a good question. As you say, there is no specific answer to that. There are some tools designed for that, but they seem to be quite stringent. We try to keep them. And since we want to continue with the protein peptides or the validation, we want to then do filtering based on the biology there. But I would do based on the coverage and poly-A signals, because this is a PCR-based experiment. But yeah, also there's specific parameters that Scanty uses that might be worth checking as well. There are many features that could be used to classify real versus artifactual.
Thank you.
Bart.
Yeah, I'm here, yes. So, thanks, elegant work. So I was wondering, you do these differentially expressed isoforms between cases and controls, or here controls and APP mice. So can you account for possible changes in cellular composition of your tissue? And is that if your cellular composition of your brains is different, does that possibly drive
We get
the
from the
transcripts you
same.
detect?
We get from the same region. It's whole cortex, but it would be interesting to see those regional differences. We haven't done it here. But yeah, done single cell as well. That's a different story. But we haven't have a specific information about the regional differences or tissue composition.
Thank you. Any further questions? In that case, we'll move on.